Healing Genes, a program of the Foundation for Cell and Gene Medicine. Three friends lounge together on a sofa and armchair. All are accomplished young men, and all were born with inherited forms of blindness. Christian, seated in the middle, is pursuing his dream of a career in music and recently released his first single. He has received the one gene therapy that has been approved for blindness. Mitchell and Michael on the left and right of him are brothers. Mitchell has completed his first year of college, and Michael graduated summa cum laude in the spring of 2022 and will be working for Disney World. Genetic therapy for their form of blindness is being pursued but not yet ready for clinical trials. The three men describe their hopes for gene therapy. Michael speaks first.
So for me, it would be extremely useful and more efficient if I were able to do things like read signs and move through airports on my own.
Christian.
And because a gene isn't there, it's not producing the proteins needed for your cells to live, so they all slowly start to die off, and that causes the inevitable blindness. But really, I just wanted to stop that.
Mitchell.
Even something simple like I use a white cane, and to be able to have that extra hand open, you know, just to do things like carry things, whatever it is, you know, I just, all around seeing is a good thing.
Genetic therapies are being developed and used to treat inherited forms of blindness. These therapies use viruses that don't cause disease, but are good at carrying things into malfunctioning human cells. The therapies use what are called adeno-associated virus vectors that either carry in working genes to replace defective genes or carry in molecular scissors, a tool that can basically make a cut-and-paste edit of our genetic code. One therapy that replaces a defective gene has been approved by the Food and Drug Administration. However, the genes for some forms of inherited blindness are too big to fit into the virus. So researchers have determined how to slip in a tool, the molecular scissors, that can make the needed changes to the genetic material directly inside the cell. One such trial has begun. In both therapies, many copies of the virus are loaded with their cargo, either the missing gene or the molecular scissors, and placed in a syringe for injection, as shown here. The therapy that has been approved by the FDA is for people born with a defect in the RPE65 gene. The defective gene results in many cells in the eye's retina not producing a protein needed for sight. In this illustration of a DNA helix, the defective gene is shown as a faded-out section. In this therapy, working genes shown in bright green are inserted into the virus and replace the defective genes. Many viruses are employed to carry in the working genes. The gene-carrying viruses are drawn up into a special syringe. The target of the viral gene carriers is the retina in the back of the eye. The viruses are injected directly where they can get into cells called retinal pigment epithelium that are critical for sight. In an illustration of these cells, the defective gene is faded out to indicate it's not working. Once the virus delivers the working gene, shown in bright green, to the retinal cells, it can make the protein which is needed for sight. After Christian received this therapy, he could see the moon for the first time. A therapy using the genetic scissors known as CRISPR is in a trial now for another form of inherited blindness. And a therapy using CRISPR for the genetic defect that Mitchell and Michael have is expected to be in clinical trials in a few years. The CRISPR therapy being tested now is directed at correcting a defect in the genetic code of the CEP290 gene. This gene, when defective, causes blindness because a needed protein is not being made. In this illustration, the defective gene is faded out, indicating it's not making the needed protein and often resulting in severe early onset impairment of vision. Once delivered by the virus, the CRISPR technology, represented here by two sets of scissors, cuts out the bad letter in the genetic code, allowing the gene to work normally again. Once the CEP290 gene can work again, the eye's retina can produce the protein needed for sight. Early results from the clinical trial show some patients regaining some sight. Catching up with the three friends we met earlier, gene therapy did stop the inevitable decline of vision for Christian. Christian's musical talent scored 1 Golden Buzzer on America's Got Talent, and he was in the top 7 finalists. Mitchell and Michael both have high hopes for the CRISPR therapy. Michael will be working for Disney World. Mitchell is studying communication in college, and as he told us,
I can at least try and get some of it back. And I am really in the boat of trying to get it back.
Thank you, Michael, Mitchell, and Christian. This program was produced by the Foundation for Cell and Gene Medicine, narrated by Michelle Spitz. Music, Spirit of Success by Illumo. Audio description produced by Cliff Hahn Sound. Animation and editing by Dragonfly Media Group. Copyright 2022, Foundation for Cell and Gene Medicine.